am Madhurima Datta, a zoology and biology educational content creator. Now's content for B.Sc and M.Sc in zoology honors, microbiology honors, physiology honors, biochemistry honors, biotechnology. An exam for CUET PG exam, GATE exam, DBT exam, IIT JAM exam and CSI NET Life Sciences exam. Paper Genetics and Unit 1 Introduction of Genetics. Today I will discuss about the law of independence. In topic, the law of independence of Mendel's laws, first I will discuss about the dihybrid process. What is the dihybrid process? Mendel also performed experiments with plants that differed in two traits. He crossed plants that produced yellow round seeds with plants that produced green wrinkled seeds. The purpose of the experiments was to see if the two seeds traits, color and texture were inherited and independently, because the F1 seeds were all yellow and round and the alleles of these two characteristics were dominant. Mendel grew plants from these seeds and allowed them to self-fertilize. He then classified the F2 seeds and counted them by phenotypes. Next, description of the process. The four phenotype classes in the F2 presented all possible combinations of the color and texture traits. Two classes, that is yellow round seeds and green wrinkle seeds, which resemble the parental strains. And the other two, that is green round seeds and yellow wrinkled seeds, show new combination of the traits. And the four classes had an approximate ratio of 9 yellow round, 3 green round, 3 yellow wrinkled and 1 green wrinkled. So ratio is the 9:3:3:3:1. Next, the experimental level. In the parental generation, round yellow seeds crosses with the true breeding wrinkled green seeds and then produce the two gametes, that is capital RY and small ry. Capital RY denotes round and yellow seeds and small ry denotes green and wrinkled seeds. Then produce if a gamete in F1 generation, that is capital R small r and capital Y small y, that is round and yellow seeds in the F1 generation. This is because capital R dominant of small r and capital Y dominant of small y. Then self-processor happen, that is capital R small r, capital Y small y into capital R small r and capital Y small y. And after the self-pollinations or self-crosses, they are produced. Our gametes are capital R, capital Y, capital R small y, small r capital Y and smaller small y. Then it is a self-crosses after produce capital R, capital Y, capital Y. There is 9:3:3:1 because 9 types of seeds, they are round and yellow. Then 3 types of the seeds are green and round, and then 3 types of seeds are yellow but wrinkled, and 1 types of seeds are, that is wrinkled and green. So there are produced total 16 offspring and their ratio is 9:3:3:1. And 9:3:3:1, this ratio is the phenotypic ratio. States of law of independence. The law of independence assertment states that the allele pairs for different traits segregate independently of one another during gamete formation. As a result, the inheritance of an allele for one gene does not influence or dictate the inheritance of an allele for a second gene, which provided the genes are located on different chromosome pairs or far apart on the same chromosome. Next, cytological basis and mechanism. Meiotic stage occurs during metaphase 1 and anaphase 1 of meiosis. Next mechanism, homologous chromosome pairs line up randomly at the metaphase plate The orientation and subsequent separation of one homologous pair toward opposite poles occur entirely independently of all other pairs. Next, gamete recombination. For an organism's heterozygous for small n genes on different chromosome independent assortment produce 2 to the power n genetically distinct types of gamete in equal proportions. Next, non-Mendelian deviations and exceptions. Independent assortment does not apply universally across the genome. Key exceptions include genetic linkage. Genes located close together on the same chromosome tend to be inherited as a single unit rather than assorting independently. Recombination frequency measured in the centimorgans, that is cm. An independent assortment produces a 50% recombination frequency and linked gene shows less than 50% frequency. Next, gene interactions or epistasis alters the phenotypic ratio, that is 9:7, 12:3:1 and 9:3:4 due to functional interaction between non-allelic genes through the underlying alleles still assort independently at the chromosomal level. We will discuss about the epistasis in next slide. Next, organellar or material inheritance. Mitochondrial and chloroplast genomes are inherited non-monogenically through a single parent, that is usually maternal, and bypass meiotic assortment entirely. Next, molecular and cytological basis. Independent assortment is fundamentally a consequence of chromosomal, that is alignment during metaphase 1 of meiosis, random bivalent orientation. During metaphase 1, maternal and paternal homologous chromosome pairs line up randomly along the metaphase plate. The orientation of one bivalent pair has zero mechanical influence on the orientation of another pair. Next, physical separation. In anaphase 1 and homologous chromosomes separate toward opposite poles. The physical segregation of alleles for gene A on chromosome 1 occur independently of the segregation of alleles of the gene B on chromosome 2. Next, the Punnett square method. For situation involving one or two genes, it is possible to write down all the gamete and combine them symmetrically to generate the array of zygotic genotypes. Once these have been obtained, the principle of dominance can be used to determine the associated phenotypes. This procedure, called the Punnett square method, after the British geneticist R.C. Punnett, is a straightforward way of predicting the outcome of crosses. We have used it to analyze the zygotic output of the crosses with Mendel'S yellow round F1 hybrids and a type of mating commonly called an intercross. However, in more complicated situations, like those involving more than two genes, the Punnett square method is unwieldy. We will see in the figure how the Punnett square method is related to the approach to the genetic problem that uses the concept of probability. This figure is shown in the number 4 slide in this PowerPoint. Now the topic has ended. For all video of B.Sc and M.Sc in life science, biotechnology, zoology, microbiology, physiology, molecular biology, please subscribe my YouTube channel Biogenesis by Madhurima and follow my Facebook account Madhurima Datta. Thank you everyone.